welcome back to the lecture series on bioelectricity. So, this will be the 23rd lecture. So, in the last class we talked about long term potentiation, one of the models of the memory, and further I very briefly highlighted the situation of Alzheimer's or Parkinson, amyotrophic lateral sclerosis and all other neurodegenerative disorders. And we made a comparison that what happens in the case of Alzheimer's, where you have the loss of the memory, loss of identity, whereas in the case of Parkinson we talked about there is a loss of motor coordination or the motor activities. So, there is another form of memory which I highlighted in the last class, but I have not gone in depth, that is called long term depression. So, in the case of long term potentiation, what happens, there is a very high frequency stimulus, very high frequency stimulus, and that high frequency stimulus leads to a change in the network properties, so that the network it gets potentiated and it remains active for a long period of time, and during which there is permanent, it is believed that there are permanent changes within the system. But apart from it, there are situations where you do not get very high frequency stimulus. I was trying to tell you certain situations like, you are walking, you are learning certain things in a much more day to day setting, which is you are not very intense into it, but you learned eventually. So, this is something like you are getting low frequency stimulus, you are not really very very much into it. Or there are situations like fear, where you see a very small impulse for a very limited period of time, but that is good enough to resulting in a fear psychosis in your brain or some kind of anxiety. So, these things falls under the second model, which is called long term depression, LTD, and it was around 1980s. One of the Japanese called Ito, who was involved, was pioneer in starting the work on long term depression, and eventually there are several people who picked up this model. One of them is Taras Sajnowski, and in Indian context there is Sumantra Chatterjee in NCBS. These are the people who have explored it further with long term depression. So, essentially if you had to compare that what exactly happens in long term depression with respect to long term potentiation. So, I will be giving you a handout at the end of the course, where you will have a comparative picture of both long term depression and long term potentiation. But just for your understanding sake, I will kind of draw the situation which will give you an idea what essentially is happening. Okay. So, coming back, so now we are into lecture 23rd. Lecture 23rd, we are talking about the long term depression or LTD. So, before we talked about long term depression, let us see what happened in long term potentiation, LTP. In the long term potentiation, there is a very, so say for example, something like this. So, there is a very high frequency stimulus, and because of the very high frequency stimulus, at some point or other, the neural activity of a network, if this is the excitable y axis is talking about the excitability, from this baseline value, because of the high frequency stimulus, it shifted and it reaches here. This is what is happening in long term potentiation, LTP, and this is your high frequency stimulus, HFS, and this is the baseline excitation, and after a point This is the new level attained when both the systems are active. This is how you can show it graphically. I will be giving the handout which will explain it further. But in the case of long term depression, what is happening is slightly different. In the case of long term depression, what you give is essentially a something like a low frequency stimulus in the case of long term depression, you have low frequency stimulation. So, basically a circuit or a network is receiving smaller inputs. It is not receiving a huge input, as I was kind of highlighting in the case of long term potentiation. And these smaller units adds up in such a way that the network becomes active at a much more lesser threshold. Say for example, if this is the threshold where the network functions, for example, this is the threshold, the baseline threshold, this network will start functioning at a lower threshold than this. So, it is going to come down. So, essentially what does that mean? That means that the circuit becomes active at a very smaller threshold, unlike a situation. Say for example, let us take a practical situation. You are riding a bicycle, you hardly need to recall anything. You know how to ride a bicycle once you have learned it. Or you are walking on the street, you do not have to decide that you are going to put which leg first and which leg second. So, this is essentially is a situation of long term depression, where the circuits involved in this kind of motor coordination gets activated with a very smaller threshold, which you do not even appreciate or understand at that point, that you are actually doing a memory recall, but it is fairly inbuilt almost at a very lesser threshold. Same thing happens in the fear psychosis. Say for example, you have seen kind of a snake at some point, and for a fraction of a moment, but that fear has activated a circuit which gets or kind of modulated a circuit which gets activated at a very lower threshold of electrical activity. That is exactly the long term depression. So, there are areas in the brain like amygdala, which is involved in the fear psychosis, that follows the long term depression model. It is believed that this is following a long term depression model. Similarly, all your motor activity, cerebellum and further down, they follow long term depression. It is interesting, the hippocampus too follow both long term depression as well as long term potentiation. But all these different electrical phenomena, what we are discussing, are they really happening inside the brain or not, is a different question. Most of these studies have been done outside the system, where in a controlled slice or a network of neurons, these phenomena has been observed. Okay. So, in summary, if we have to say, the difference between long term potentiation and long term depression is that long term potentiation is a process where there is a high frequency stimulus, and that high frequency stimulus leads to make that particular network hyperactive, and the output of that network is higher than the baseline. Whereas, in the case of long term depression, what is happening is that from the baseline, this also becomes hyperactive, but at a much more lesser threshold. It does not need any further at a much more lesser threshold, it becomes more active. Just fraction of a moment it will become active. Okay. So, these are the two key models which ensures our, at least it is believed, are the major model for memory acquisition. And during this process, there are certain permanent changes which are taking place out here. certain permanent changes taking place here, certain permanent changes taking place here, and these changes are taking place at the molecular level or at the cellular level. Changes at the molecular / cellular level. Essentially, these changes ensures that our information gets stored. Now, from here what I will do, I will move on to the basic waves which are generated by the brain and their significance. So, in order to just let me save it. Okay. Now, by this time you have realized it is a dense network of neurons, and they are continuously involved in transferring information or consolidating information or storing information. So, your brain remains electrically active throughout your life. So, during this phase, depending on what is your physiological status at a specific time of the day or night, brain generates a series of waves, alpha waves, beta waves likewise, and these waves could be detected by placing electrode on top of your head, by which you could have multiple electrodes, you could have 64 electrodes set all over your brain, and based on those frequencies one can figure out that what activity you are doing or how much your brain is active, and likewise, and this whole thing falls under electroencephalogram. Okay. So, let us move on to electroencephalogram, EEG, for monitoring brain activity. It stands for electroencephalogram. Okay. Now, what essentially happens, so for example, what you do is that you place different electrodes like this on the head. These are just surface electrode, and what you are essentially measuring here, you are measuring these are the electrodes which are placed. You are measuring the field potential. So, at one point there are thousands of neurons, and the summation of the electrical activity, that is what you are measuring. It is a very gross measurement. You cannot figure out what is exactly happening at the individual cellular level or at individual synapse. What you are essentially getting is a pool of synapse arising from thousands of neurons at one point, and the total summation of that, and how that influences, or how that could tell you, or how you could picture the state of the brain based on those waves all over the brain. Okay. Now, what are the different waves which are involved in the brain? You have alpha waves, you have beta waves. We will come to the details of all this. Alpha waves, you have beta waves, you have theta waves, then you have delta waves. What these are? So, if you look at alpha waves occur in the brain of a healthy awake individual. Okay. In healthy, awake individuals. Okay. Whereas, very interesting, this alpha wave disappears when you are sleeping. Okay. So, let me just and it disappears when you sleep. Okay. Whereas the beta waves are typical individuals who are either concentrating on task or they are in a stress situation. So, just use another color. So, you are, say for example, you are concentrating or stressed out, you see the beta waves. Then you have the theta waves. The theta waves may appear transiently during sleep in normal adult, but are often observed in children and intensely frustrated individuals. This is a very interesting kind of situation. Mostly it is seen in children, or when a person is very, very frustrated, you see the theta waves. Whereas you have the delta waves, these delta waves are very large amplitude, low frequency waves, large amplitude, low frequency waves, and they are normally seen during deep sleep in individual of all ages. So, if you realize these are the ones which are something like, if I had to just draw a comparative picture of the different waves. So, they will be something like alpha will be when the person is awake, this is what you see as an alpha, whereas the beta is even much more like, this is the intense situation when you are concentrating or you are very, very stressed out on something. Okay. This is the kind of beta waves which are coming, and then you have the theta waves, which is person is frustrated, or you see in the children likewise. And then you have the delta waves, which are much more. So, these are the delta, these are theta, this is beta, and this is alpha. So, these are the 4 different wave patterns which are being seen in the brain. So, these are the different 4 patterns which ensures the what state the brain is, and how it is kind of altering depending on the state of the individual. So, based on this, several psychological tests are being done. Is the nature of this person, how intense this person is, and the doctors make whole lot of observations on these kind of individuals, that which wave are prevalent on which individual, and they also highlight a lot of physiological disorders. Say for example, in the case of epilepsy, you see a huge change in all these things. In the case of epilepsy, where there is basically a seizure is taking place, you see there is an absolute asynchronous behavior among all these different waves, and this is something which those who are expert in reading these waves can figure out that this person suffers from has a tendency to suffer from epilepsy, or the wave pattern completely changes over a period at that particular point of seizure, okay, or just before that it started kind of disbalancing. So, we started with in the class about, when I talked to you about that, we will be talking about how to measure electrical signal at the single cell level. We talked about how we study the different kind of channels. Then we talked about how we could pattern the neurons on top of microelectrode arrays, how we could use sharp electrodes to make the measurements. And here we are talking about electroencephalogram, which is on a live animal. You are making this field potential recordings. So, all these different kind of recordings had their advantages and disadvantages. When you do a field potential kind of measurements like alpha, beta, theta, delta waves, you get a gross picture of the brain, how it is functioning. And without any invasive technique, it is fairly straightforward. And of course, these techniques of the waves are being further assisted by the MRI and the positron emission tomography and magnetic resonance imaging. These two other techniques, very profoundly powerful technique, helps in really picturing the brain, what essentially is happening inside the brain at a specific point of time. But they won't tell you anything at what is happening at the cellular level. Whereas, if you come down at the cellular level, you can understand the phenomena at a single cell level or maybe a group of cell, but you have no idea about how the population is behaving at that point of time. Then you can further go down, you can make circuits of 1 neuron, 2 neuron, 4 neurons, 6 neurons likewise, and you can understand the small network behavior. But what is the take home message of this whole process is, this whole process are slowly divulging or disclosing those secret stories of our own self, why we are like this, why our behavior is like this, why certain people have sleep related disorder, why certain people have hallucinations, why certain people can't concentrate at certain things, whereas others can concentrate, why certain people gets stressed out, on the other hand why certain people does not get stressed out. There are a series of such things which are highlighted by by these different techniques, which eventually leads us to understand our final frontier, the brain. Okay. Now, I will give you a very brief, very brief outline about some of the sleep rhythms, what is there, and we will move on from there. Okay. So, sleep, this is a very interesting phenomenon and sleep related disorders. So, when you are in their deep sleep, basically, so there are different kind of, there is something called a REM sleep, and in a REM sleep, because this is called rapid eye movement sleep, this is where you see all the dreams. Okay. And it's a rapid eye movement, it's something like this. Okay. Then you go to the deep or slow wave sleep, and this slow wave sleep is the time when the waves are much more So, whatsoever dreams you see pretty much out here, and this is all electroencephalogram recordings. Okay. And essentially, you will observe that deep, basically, as you are kind of initial phase of the sleep, which is basically the REM sleep, and then there is a transition phase between the REM and the deep sleep. So, from the REM to the deep, there is a small transition phase, and then you will realize that most of the deep sleep in an average individual takes place between 10 a.m. to that midnight period. It's very interesting. I mean, this is where maximum deep or slow waves functions, and after that again there are rapid eye movement and likewise so on and so forth. So, one of the challenges what we are or we are going to face is we will be needing more and more tools which could couple with the existing tools of MRI, PET, electroencephalogram in order to understand or get a much more holistic picture of the brain, what exactly is happening. And more and more research is going on in the field of imaging, especially the brain imaging, to understand some of these very deep rooted phenomena of consciousness, learning, memory, sleep, dream, and the relevant disorders. And as we will be moving through, we will be talking about the man-machine interface. One small part will be dealing with it. There you will see how we really could, hallucinations, and how we could really, control the brain activity. Of course, not in the human level, but at least definitely at the levels of the monkeys. I will be giving you a certain handout, and I will be discussing that with you. Now, I will just take a small detour with this background. I will close in here with this part of the sleep, and I will come back to one of the part what we talked about the spinal cord. So, yesterday I talked about Alzheimer's, I talked about Parkinson disease, but what I haven't talked about is the amyotrophic lateral sclerosis, ALS. In the case of ALS, what happens is that, so I told you in the case of PD or the Parkinson disease, it is substantia nigra within the brain which is kind of getting affected. So, there is a region called substantia nigra. So, these neurons fail to send electrical stimulus, whereas in the case of ALS what happens? So, these neurons sends the message to the lower motor neurons which are sitting in the ventral horn. These motor neurons, when they start to die, that neurodegenerative disorder is called amyotrophic lateral sclerosis. So, essentially your substantia nigra is all fine. So, the electrical stimulus is coming from here, but from here what it is supposed to relay the electrical informations to the target tissue is not taking place. So, this is the situation with amyotrophic lateral sclerosis, and further there are situations when, because of accidents or because of some kind of mess up, basically these neurons started to die or dies out, certain part of the circuit kind of get damaged, which is essentially spinal cord injury. This is which mostly happens during automobile accident or some other very hasty situation. So, in the case of spinal cord injury, there are several routes. I mean, one of the possibilities of future will be some way or other, if we could regenerate those neurons which are kind of damaged at a specific part. Okay, you implant the stem cells, and they somehow or other get incorporated, or you can put a neural chip out here, something like neural chip which will take care of it, or you could have an artificial device which will rebuild the connectivity between. So, it is almost like a fuse. Okay, the fuse goes off, then this connectivity has to be reestablished, or you follow a stem cell therapy, or somewhere or other you do put certain stimulating electrodes which will help to regain or maintain some of the activities. Okay. So, there are several techniques at the level of neuroprosthesis. So, this is all regarding spinal cord injury situation. Okay. So, there are several techniques by which people are trying to counter the spinal cord injury patients. Similarly, on the same line, if I go back to the brain, So, for example, what will happen if part of the brain dies out, something like this. Say for example, out here there are people who are trying to implant chips on the brain. Attempts are going on that if we could implant a chip on the brain at the place where the damage has taken place. Of course, the other therapy will be you incorporate stem cells out there, and those stem cells become electrically active. First of all, they become neurons, and those neurons become electrically active. So, it means whenever we talk about electrically active, that means essentially they should be able to generate their sodium channels, and then they get incorporated in the circuit. But we do not know really if these such cells get incorporated into the circuit, how the connectivity will be dictated, what kind of connectivity they will establish with the rest of the circuit where the previous cell has died. So, just to visualize the situation, it is something like this. Say for example, you are in a network like this. Okay. These are the individual neurons what I am drawing. So, say for example, this neuron dies out, this one dies out, and say this one dies out, and these. So, now instead of them, here you are putting some new neurons which you are assuming they will be they will form neurons, some stem cells, but will they form the same connectivity or not is a big question. So, these are the challenges of the future. Will they become neuron? Will they express all the necessary ion channels to become neuron? We really do not know at this stage. We are just speculating, yes, it may happen, it will be helpful, but we really do not know. Or could we put certain electronic device out there which will get incorporated and re-establish the connections? We really do not know. These are some of the final frontiers where mankind is kind of or dreaming, struggling, and moving towards to understand who we are. So, at this stage, after covering through the central nervous system, peripheral nervous system, briefly talking to you people over the last 2 classes about the memory, learning, sleep, very briefly about the sleep, and giving you an overall picture, I will close this part of the talk, and in the next class I will touch few smaller topics which I missed out in the course, which will make much more sense at the cellular level, especially in terms of the neurotransmitter, excitatory, inhibitory circuits, and that is where we will be closing down on the animal bioelectricity. Okay. Thanks a lot.